Hello everyone and welcome back to another episode of the World of Sharks podcast. You guys know the drill by now. This is a podcast all about sharks, rays and their underwater habitat, brought to you by the Save Our Seas Foundation. I'm your host, Isla. I'm a scientist and a science communicator for the Save Our Seas Foundation, and I love talking about sharks, which is a very good thing because my job is to interview experts in shark and ray science, conservation, education, and storytelling to take you on a deep dive into a different part of the wonderful world of sharks. This week, we are zooming in to find out just how much we can learn from shark DNA. And we are very lucky to have an amazing expert to walk us through the field of shark genetics and genomics, Professor Mahmood Shivji. Mahmood is a professor of marine science at Nova Southeastern University's Oceanographic Center in Florida, and he is also the director of the Harvey Research Institute and Save Our Seas Foundation Shark Research Center. He leads both the research and education programs of the Shark Research Center, which specializes in taking integrative, multidisciplinary approaches to research and conservation. That includes combining state-of-the-art genetic techniques, genomics, and fieldwork to better inform conservation management strategies for endangered species of shark and ray across the world. Mahmood is a world-leading geneticist who is behind some very important discoveries that have been made in the last 2 decades relating to shark and ray science and their conservation. For example, his pioneering research developing rapid DNA forensic methods to identify shark body parts is still being used by the US and other national fisheries management agencies to reduce the illegal fishing of threatened species of sharks. He was also part of the first research team to sequence the entire genome of the white shark, and more recently this year, the entire genomes of both the shortfin mako and the great hammerhead. Don't worry, we will explain what all of that means in this episode. And this episode also has something for everybody. So if you know absolutely nothing about DNA, about genes, genomes, genome sequencing, we break down all of these concepts. Well, I say we, Mahmood breaks down all of these concepts. And if you're like me and you kind of know a little thing or two about genetics from school or from university, we dive a little bit deeper into the research of Mahmood and his team and discuss kind of what this means from a conservation perspective. It's got a little bit of something for everybody. So, without further ado, grab your lab coats and let's dive in to our episode. Hello, Mahmood Shivji, and welcome to the World of Sharks podcast.
Well, thank you, Isla. It's a pleasure to be here.
Thank you so much for coming on. I'm very, very glad that we are able to do this. I know how busy you are, and we very much appreciate the time that you're taking to walk me through, not just the listeners, walk me through the very fascinating but also incredibly complicated world of shark genetics. But we have the perfect person to lead us through here. So I'm very, very excited to have you on. But before we dive in to those questions, we have some, I hope, slightly easier ones just to warm us up a little bit. And we like to start the podcast with the same question for every single guest. And I know it's quite hard when you've been working, in the marine space for most of your life to pick out an experience that, stands out among the rest. But is there an experience that you have had with the ocean or adjacent to the ocean that stands out for you as most memorable?
Well, I have been working in the marine environment for a long time, as you mentioned, Isla, and there have been a few such occasions. But I think my first such standout memory goes back to when I was about 20 years old and working as a research assistant in what at the time was a very isolated place on the west coast of Vancouver Island in Canada. And we had a team of 6 people. And one day, just for fun, we decided to go diving on a shipwreck that was really quite far away from our research lab. And it took us about 4 hours to get to the shipwreck in our really small inflatable boats. They're about 5 meters in size. And that journey was an adventure in itself, given how far we had to go across choppy seas to get to the shipwreck. And on the way there, we encountered a lot of gray whales and even had one whale breach in between our two boats, which was quite a sight. But really, that wasn't the most memorable experience. To top it all off, when we got to the shipwreck and dove in, that's when I had my first mind-boggling diving experience. And the visibility was pretty good. Actually, it was very good, if I recall. And you could see the wreck from the surface down to about 30 meters. And as we went down, as we descended, we were surrounded by hundreds, many hundreds of small sharks called spiny dogfish. And it was like descending through a cloud of sharks. And these sharks kept on bumping into us and then into the divers and then turning away the whole way down as we descended onto the wreck. And it wasn't an aggressive behavior at all. It was just like, we were in the shark's way. And amazingly, once we got to the top of the wreck, there were no sharks there. So they all stayed just right above the wreck. And so we had descended to this cloud of sharks. And then we get to a place on the wreck where there are no sharks. But if you look up, there were these hundreds of sharks the whole time. And that experience is something I'll never forget. I can still picture it. And unfortunately, in those days, underwater cameras were huge and very expensive, no GoPros. And so no one had a camera with them. And so we didn't record any of this, but it's made an indelible impression on my brain. And I still think about that sometimes. It was an incredibly memorable experience.
Yeah. I can completely see why that stands out in your mind. That's absolutely incredible. And one of the things that I've always wanted to see are, the very similar thing that, we're going to come on to a little bit later on, but there's the scalloped hammerhead aggregations in the Eastern Tropical Pacific that look very similar to that. So like you dive down, fairly deep and you look up and there's just a whole ceiling of sharks going overhead. But as a cold water diver, to think of that with spiny dogfish is very, very cool and very exciting. And that's now on my list of things to see. But you credit your fascination and your love of nature and the sea to growing up somewhere that's very far away from Vancouver Island in Kenya. Can you tell us a little bit about your childhood and how that early connection to the ocean formed?
My family lived on a small hillside overlooking an inlet on the island of Mombasa, which is the very, very southern coast of Kenya. And as kids, we would go to the inlet and swim and play in the ocean quite a lot. It was just something that, we did. In fact, we didn't have television in those days. And, no, that's not quite right. There was television. It was just that my family didn't have a television. And so we just played outside a lot. And the surface there of the ocean was also a mangrove habitat. And so we would see all kinds of marine wildlife. But we were just kids. We were just playing. And I wasn't really thinking about wildlife per se, but I'm sure that those early experiences had something to do with developing my fascination with marine biology and marine organisms. Now, interestingly enough, for university, I didn't start off studying biology or marine biology. I started off studying economics for a first degree. Then it turned out that we had to take a science class as a requirement, just for an economics degree. And I took a biology class, and I just found the material fascinating. And so I think the combination of my childhood experiences playing in the ocean and seeing lots of amazing ocean life, and then becoming aware of biology as a formal discipline later, sort of all came together in my brain and sent me off in the direction of marine biology.
And you were like, this is the one for me.
Yeah, Not that I didn't like economics, I actually did, but I just remember being so amazed and fascinated by learning about biology of a wide diversity of things in that first class, I think.
And then how did that lead you, or how did you get into shark science then?
Well, my involvement in shark science was completely serendipitous. Even though I had that experience with all those spiny dogfish and diving, when that amazing experience there, I was really never thinking about doing anything with sharks. I started my position as a professor here at Nova Southeastern University's Oceanographic Center, which is still the university where I am currently. And because it was an oceanographic center, I had to work on ocean-related topics, but I was trained as a geneticist also. So I was looking to combine marine biology and genetics together. And in those days, very few people were doing DNA research on marine species, although now it's quite a big field. And the connection to sharks specifically, or shark research for me, occurred when I was reading the local newspaper. And I saw an article that U.S. fishery managers were having trouble identifying shark body parts that were being brought to port by fishing vessels. And by body parts I mean, certainly fins, as well as carcasses that, where there's no head or tail or fins were all taken off. And they call them logs. And they were having trouble identifying them because many of the fish species looked very similar to each other. Things like silky sharks and dusky sharks and blacktip sharks and Caribbean reef sharks. These are all sharks in the genus Carcharinus and have very subtle morphological distinctions. So you really have to know what to look for to tell them apart. And then when you take the fins off and the head off and the tail off, then it becomes even more difficult. And because of that difficulty of identifying these body parts, fishery managers could not keep track of how many sharks of each species were being actually brought to port or being landed. And I read about all this in the local newspaper, that the fisheries managers needed those identification data to keep track of how many of each species were being killed, so they could properly manage the fisheries. Now, I wasn't thinking about sharks really, but as a geneticist, I thought, oh, well, hey, I can solve that problem simply by looking at the DNA of these shark body parts to identify species. And so I contacted the management agency, some scientists at the management agency, and asked them to send me samples from sharks that they knew the identification of, and they were very kind, and they did that. And I started working on developing these rapid genetic methods to identify shark species just from a small piece of meat or fin using their DNA. And that worked out quite well. And that sort of genetics angle, the genetics pathway, led to my entry into the shark research world. So completely unexpected. And I can thank a newspaper article in the local press.
But what was it, so excuse the pun here, but what was it about genetics that kind of hooked you in?
That was really entirely the fault of my academic mentor. His name was Professor Michael Neuschel. When I was a master's degree student at the University of California in Santa Barbara, and really I was in his lab, and I was studying ecology, kelp bed ecology actually. And Professor Neuschel tried right from the start, I remember this distinctly, to try to convince me to include genetics into my research. But I resisted. I just, didn't know much about genetics, and I just wanted to be a field ecologist. But, it was only at the very end of my time as a master's student that I finally understood the power of genetic technologies to dig into the very core of understanding biological function, regardless of species. So all thanks to my former professor for steering me to a field that has now just exploded with mind-boggling technology and discoveries. A geneticist can work on any species, right? From bacteria to humans to fruit flies to elephants and whales or anything, to coconut trees. And of course, sharks. So it's a versatile field and an incredibly exciting field nowadays with these amazing technological innovations that are coming out for DNA sequencing.
It must be a super exciting field to be part of and to have seen flourish as well, because it kind of really is. I mean, correct me if I'm wrong, but it kind of really is in the last decade or last 2 decades that it's sort of really, taken off, and there's been a lot of advancements to the field. And I like it as well that you said when you first started to describe how you got into the field of genetics, you said it was the fault the fault of the supervisor. Not thanks to him, it's his fault.
Very wise chap. He had real vision. I mean, he saw the future, long before, and tried to steer me into it long before it became popular to do genetics. Yeah.
Okay, so before we fully plunge ourselves into the waters of genome sequencing and DNA sequencing, I wanted to take us right back to the basics of genetics, because I say basics, there's nothing basic about this field, but there might be people listening who we are going to go into a lot of terminology, like we're already talking about DNA, about genes, we're going to be talking about sequencing, and I wanted to just kind of rewind a little bit and explain what some of these concepts are, because there might be some people listening who have either never heard of it before or who, like me, have, remember these terms vaguely from school or university, but never actually went on to study them and don't know them as in depth as you do. So I wondered if we could go right back to the start and kind of begin with DNA. So I've always imagined, or if I remember correctly, I always think of DNA as the basic instructions for every living thing. So kind of, if you imagine that you're a piece of flat pack furniture and DNA are the instructions that basically dictate what you're going to look like, how your body functions, and then genetics, in a very simplistic sense, is basically learning how to read these instructions and figuring out how these instructions have led to the actual complete living thing that is in front of us right now. Have I got that right, or am I completely butchering your field?
You're exactly correct. It's the basic instructions of how life works, and these instructions are coded for in DNA, and then also how those particular traits are passed on from one generation to another. So yeah, that is genetics.
And then, of course, with the Shark Research Center, the DNA that you're looking at and that your research team are focusing on comes from sharks. And I was interested to know kind of what kinds of things can we learn from a shark's DNA. I just find it so fascinating because obviously we can't see DNA with our naked eye. We can obviously see the products of it, but we can learn so much from it. So what can the shark's DNA tell us?
Okay, so shark DNA. I mean, DNA, whether it's a shark or a coconut tree or a fruit fly, is still a very simple molecule, right? Most people will remember that it's just these DNA is made up of these 4 bases, the A's, T's, C's, and G's, and there's long strings of these things. Now, the DNA in an organism, whether it's a shark or anything else, is made up of many different sections or different segments, some of which, it's all just DNA, A, T, C's, and G's, but some of those sections are actually genes. But a lot of the DNA in an organism is made of sequences that control the expression of those genes. So they're not genes themselves, but they control how those genes are expressed and what proteins are produced. Now, these DNA sequences can tell us many, many things that underlie biological function regardless of species. Now we happen to work on sharks, but the principles are the same. And so the genetic technologies used are the same, whether one is studying humans or one is studying sharks. Now, a lot of the technology with DNA has been driven by human medicine. And for humans, the main driver of DNA sequencing based genetics research is understanding the causes, the inheritance, and treatments of human diseases. And this business, this human genetics business, is now a massive industry. But DNA sequences are also used to study human ancestry, historical migration patterns, and in crime forensics. Where solving crimes using DNA is now a routine practice, and there's lots and lots of TV shows about this stuff. Now, for sharks, and actually all wildlife, analysis of DNA is used to discover many different aspects of their biology, some of which are directly related to conservation. So, for example, at the Save Our Seas Foundation Shark Research Center, we use DNA analysis for identifying different shark populations across their range. Some shark species have global ranges, and this is really important for conserving different genetic stocks of sharks. We use DNA for getting information on shark migration patterns. Many of these sharks travel huge distances. We use DNA analysis in shark forensics, such as identifying shark species from very tiny amounts of their body parts in commercial markets, as I explained earlier, to see if the sharks being sold are from legal to catch species or endangered and protected species. We use DNA for estimating shark population sizes from their DNA. We use DNA for understanding shark behavior in terms of their mating systems. For example, we were the first ones to show that female sharks have this amazing biological property of being able to produce progeny, babies, by parthenogenesis, or virgin birth. So that is without the eggs of a female shark needing to be fertilized by sperm from a male shark. That was really big news when we published that discovery. Nowadays, we are also using DNA analysis to sequence the entire genetic makeup of sharks, their genomes, to understand what makes them these amazing biological animals, organisms really that they are. So fundamentally, DNA is being used by us, and we've been in this business for quite a long time. But now many other researchers that are studying sharks.
There's so much to learn, and we're kind of going to explore some of those questions in more detail in just a little bit, kind of looking at some of the big publications that the Research Centre have published, even just this year, to kind of illustrate your points there. And one of the biggest achievements from this year that your research team were able to do was to sequence the entire genome for two endangered species of shark, the shortfin mako and the great hammerhead. This was work led by yourself and Michael Stanhope from Cornell University. And you, as I said, you managed to sequence the entire genome. So we just described there what the sequencing of DNA is. But what are we referring to when we say, a whole genome for a species?
Right. So a whole genome is basically the entire genetic content of an organism. It's very simple. I mean, you take all the DNA in the species, that's called the genome. And the genomes of all species, including sharks, is made up of DNA. So when you sequence the genome, it just means obtaining the linear order of these four basic building blocks, or the four DNA bases, the A's, T's, C's, and G's that make up the DNA content of the entire genome, of all the DNA content in an organism. Now, getting the DNA sequence of the whole genome, genomes tend to be very large, entails multiple steps. And the use of still, pretty expensive DNA sequencing equipment Although, luckily, the sequencing cost per DNA base is coming down really fast as the chemistry, the sequencing chemistry, and the sequencing machines get better and better. Now, as far as shark genomes go, what is interesting is that many sharks have genomes that are at the higher end of the size scale among vertebrates. For example, we found that the genome of the short-fin mako that we sequenced recently, that you mentioned, is a little more than 1.6 times larger than the size of the human genome. The great hammerhead genome, on the other hand, is a little bit smaller than the human genome. And, these are things we weren't expecting, but that's what the genome sequencing told us. Now, the bigger the genome, the more sequencing it requires. And so the cost of getting a shark's entire genome, especially big genomes like makos and white sharks, is unfortunately higher than, say, sequencing a human genome, at least for now. Just because there's just so much more DNA to sequence in many of these sharks.
Yeah, so it's kind of like, if we go back to a kind of like instructions analogy, it's like if you're sequencing DNA, you're kind of looking at the individual instructions themselves, whereas sequencing an entire genome is basically like getting the whole manual and trying to put that together from the beginning.
That's a perfect analogy. In fact, if you don't mind, I'd like to use that next time I talk to somebody about this.
You can use that if you want. Just quote me. Cite me. Yeah, no, but that's the kind of the way that my mind visualizes it, and I can imagine, people kind of they've got the human manual, and then someone hands them the short fin mako one, and it's just, so much heavier, and you're like, oh my goodness, that's going to take me quite a long time to decipher. It's like almost like building, the world's biggest flat pack furniture, trying to decipher it in a foreign language. What did you learn from, sequencing the genomes of those species, and what does that mean from a conservation perspective?
So, okay, so we sequenced the genomes of the great hammerhead and short-fin mako shark recently. We published that earlier this year. But the first shark genome we sequenced was that of a white shark, and we published that in 2019. We discovered that not only are white shark genomes, also about 1.5 times the size of a human genome, so big again, but it also showed that white sharks have some amazing genetic adaptations for keeping their genomes stable. Now, what is genome stability, right? It's a bit of a stable. Stable just means things that is not changing very much, and genome stability, turns out, is actually a good thing in an evolutionary sense. In that, what it means is that white sharks, having these properties of having a stable genome, means that they are less susceptible to the many diseases, such as cancers and neurodegenerative diseases that occur when genomes are unstable. Unstable genomes simply means that a lot of mutations that are occurring. So stable genomes means there's less mutations occurring. Now, in addition to finding that white sharks have pretty stable genomes, we found that the DNA sequences of genes in white sharks that are known to be involved in wound healing, the white shark genes involved in wound healing, are also very efficient. They function in a very efficient manner. In a way that was very interesting, because all shark biologists that work in the field will tell you that sharks can heal from wounds really, really quickly. And part of it makes sense as an adaptation, because when sharks mate, males grab onto females. They don't have hands to do it with, and they use their teeth. And shark teeth leave huge wounds in female sharks during mating season. And when you look at these fresh wounds, you sort of wonder how these females even survive. But they heal really fast. So there is an underlying genetic mechanism that is allowing them to heal really fast. And we found the genetic mechanisms by sequencing the genome of a white shark. So That was our first foray into the world of deciphering shark genomes and looking deeply into their biology on the basis of their entire genomes. So really, we cut our teeth, really, if you will, on the white shark in terms of genome sequencing. We learned a lot. We made a lot of mistakes, but we also, figured out how to do it by the end. And then by the technology of sequencing had improved also, and then we sequenced the genomes of the great hammerhead and the short-fin mako for the first time, which we then published early this year.
And in terms of like the short-fin mako and the great hammerhead genome, I know that we, learn all that fascinating stuff about white sharks and their abilities to heal from injury. Did we learn anything similar about the hammerhead and about the mako from their genomes?
Right. So for the hammerhead and mako, we did not, so far, haven't looked at the genome stability and wound healing aspects. We focus our attention more on conservation aspects because ultimately, our funding is largely comes to do conservation research. But once you have those genomes, then you can dig into them at a variety of different levels to answer all kinds of questions. For the great hammerhead and the short-fin mako genome, so in addition to just providing these really high-resolution chromosomal level genome characterizations, so now anybody can take that and dig more into them and find more things. But a big part of what we did with these genomes was we looked at some conservation genetic aspects, like the conservation genetic status of great hammerhead and short-fin mako compared to other endangered species of all kinds, like pangolins and tigers and wolves, etc., whose genomes have also been sequenced. So we wanted to see how the genomes of the great hammerhead, which is critically endangered according to IUCN Red List, and the mako, short-fin mako, which is endangered, compared to genomes of these other land endangered species. And what we found from this particular viewpoint, conservation viewpoint, was both optimistic and pessimistic, depending on the species. For example, genomes reflect the current genetic status of species. And what we found in the great hammerhead genome were these really long stretches of DNA sequence that had very, very low genetic variation, surprisingly low genetic variation, and overall very low genetic diversity across the whole genome. In fact, a great hammerhead genetic diversity across the whole genome was one of the lowest compared to even endangered land animals. And from the genomes, from the genome sequences, we also found evidence of very high levels of recent inbreeding. And inbreeding is never a good thing when it comes to biological fitness and adaptability to changing environments. And really, a lot of inbreeding makes species more susceptible to extinction. And we found that signal in the genome of the great hammerhead. But optimistically, the genome of the short-fin mako reflected much more standing genetic variation and very, very little indication of recent inbreeding, which really means that their population sizes are large enough to prevent inbreeding, which is a very good sign for the conservation status of short-fin makos.
Interesting. And this very much links us on to another piece of research that I wanted to discuss with you, which was led by Sydney Harned, but you were involved also in, the research team, which looked at the iconic aggregations of the critically endangered scalloped hammerhead in the Eastern Tropical Pacific, the ones that we were talking about right at the beginning of the podcast, that sealing of sharks. And, this, we'll put links to all of these papers and all these publications in the show notes so people can go and read them. There'll also be links to, our articles that we've written about them that kind of break them down a little bit more. And they cover so many different aspects, and there's lots of things to discuss with these papers. We need an entire other podcast episode for that. But one thing that paper also touches on is the genetic health and how it actually showed that the population in the Eastern Tropical Pacific had a higher genetic diversity than other populations that are found around the world. And I wanted to dive into that in a little bit more detail, is this concept of genetic diversity and, why that's so important to understand when it comes to things like a species genetic status and what that could possibly mean for how they will cope with different environmental conditions. Why is genetic diversity, why is that important? And why is a low genetic diversity a bad, quote unquote, a bad thing?
So a fundamental principle in biology is that high genetic diversity in a species is a very important property when it comes to survival, when it comes to the adaptability of the species to changing environmental conditions. And we have environmental conditions with climate change in particular are changing very, very quickly. And genetic diversity, or having genetic diversity, allows a selection on specific genetic groups within the species to accommodate changes in the environment, to be able to adapt to those changes and survive. And also having high genetic diversity reduces the chances of inbreeding, like we found in the great hammerhead. If you don't have much genetic diversity, and this is not just for sharks, this applies across all life forms, it reduces the chances of the species being able to adapt to environmental changes that are going on. So in the world of conservation, trying to maintain high genetic diversity in species is a really, really important endeavor. So having management plans that will maintain high genetic diversity is a big goal of conservation. But you have to know, first of all, what kind of genetic diversity there is or there isn't. Now with scallop hammerheads, there's these iconic animals that are actually, their fins are very highly valued in the fin trade, unfortunately. In the Eastern Tropical Pacific in particular, there is a huge amount of overfishing that's going on. It's called IUU fishing, illegal, unreported, and unregulated fishing. And scallop hammerheads are captured quite a lot there. And so it's certainly been a huge concern that this overfishing is reducing population sizes to the point where there is very low genetic diversity left in this species in that part of the world. So we compared, my former student, Sydney Harnett, compared the genetic diversity in scallop hammerheads from the Eastern Tropical Pacific to scallop hammerheads from other parts of the world. And we were really expecting to find very low genetic diversity in Eastern Tropical Pacific scallop hammerheads compared to other places. But actually, much to our surprise, it was the opposite, which is really a relief. And so that means that with more conservation efforts of scallop hammerheads in the Eastern Tropical Pacific, including reductions in fishing, it's a big thing, the scallop hammerheads in this region have a better chance of bouncing back and adapting to environmental changes that are rapidly occurring. So that was a good news sort of study, really, a good news discovery, I should say, from that study.
Which is nice because, we've got to cling on to all of the, the positives that we find and all of the good news that we find, because it can feel a little bit like there's just bad news constantly all the time. And so it is good to hold on to these things. And I guess just to go back to diversity, just so that I have it straight in my brain. So it's almost like, so if you have a higher genetic diversity, there's more chance of you having a gene that will allow you to adapt to different conditions. Is that correct? So it means that the population in general, there's more chance of individuals in that population surviving and being able to then breed and pass on those important genes that will allow the species to adapt in the future.
That's exactly correct. So when you have genetic diversity, and say you have some unforeseen change in the environment that kills off a bunch of individuals in that species because those individuals simply don't have the genetic makeup to survive those changes. But there are other, if you have genetic diversity, there are other individuals of that species that have the genes that allow them to adapt to those changes that are going on. And so, as you just mentioned, those individuals will then pass on, they will breed and then pass on those genes to their progeny, which will allow them to survive in under those particular conditions. So yes, so having genetic diversity is of critical importance in adaptability and therefore survival of species across the board, not just sharks.
It just amazes me how much we can gain from looking at something, you mentioned, a relatively simple molecule that is just the building blocks of life. And there's just so much that we can learn from that and are still learning. This is a really rapidly advancing field with new techniques and methodologies coming up all the time and being discovered and being developed. And I was interested to get your take on if there's anything, in relation to sharks, because this is a shark podcast. And, it goes, you've said many times throughout this, is that, anything that you can say about shark DNA can be applied, to species all over the world. But is there anything that you're particularly excited to see develop?
The answer to that is yes, and I'll tell you that the technological, and I mentioned this earlier, the technological innovations in DNA sequencing, together with all the computational analytical methods being developed for making biological sense out of these huge volumes of DNA sequences being generated nowadays because of all these incredible machines, makes it a really exciting time to be involved in our case, in shark genomics. Genetics, DNA underlies how all life functions. And now getting all of this sort of really high resolution, highly detailed information about DNA at the level of sort of whole genomes is allowing us, in addition to understanding how, in this case, sharks actually work, because, as scientists, we're also interested in basic biology, how organisms work, but also from a conservation perspective, all this genomics information is telling us, like we find out with the short-fin mako and the great hammerhead, which species need to be prioritized for more conservation efforts. And so genomics is, in addition to just basic biological information, is also a great new tool and approach to help guide conservation efforts. And so this is a really exciting time. And the technology is just getting better and better really fast.
Yeah, it's very, very exciting. And yes, I mean, an hour has gone by so quickly, and I'm so thankful to you, Mahmood, for, breaking down a very, very big, and there's so many difficult concepts within this field to, we've just scratched the surface of the different routes we could go down. And on this podcast, we probably will go down some of those tangents in different episodes and explore these concepts more. But as an overview, this has been absolutely fantastic. And I'm so grateful for your time and for your knowledge and your expertise. I say this on every episode, but I could honestly talk to my guests all day and just continue to pick your brains and ask you lots of questions, but I'll not do that. But we've talked about all of this, very interesting and very, deep science. And now I almost feel bad asking you this final question, which is a very, well, some people do find it profound, but it's a very surface level and a very fun question that we just like to end the podcast with. And that is, if you could be any species of shark, ray, skate, or chimera in the world, what would you be and why? And we've had to add chimera to that after having so many deep sea specialists on in the past. So,
right. Oh gosh, I'll go with sharks because that's what we study for the most part. We study lots of different species, not just at the genetics and genomics level, but also we do a lot of other sort of ecology work where we look at migration patterns. And I would have to say that if I had to choose one, I would probably go with the short-fin mako. And my reasoning for that is partly because I've been diving with them, and I see, they're like torpedoes. They look like torpedoes. They've got this incredibly pointed nose, these huge eyes. They can swim enormously fast. They make these incredible leaps out of the water. They're an impressive high performance animal, and their genetics seems to be pretty good so far anyway. So I think I'd go with short fin mako.
You're gonna pin your bets on the winning team.
Right.
And they are. I mean, that's not the only reason. They are an extremely cool species, and yeah, they can move fast. They can travel, great distances and go deep and go shallow. Yeah, I think short fin mako is a very, very good answer. Yeah, I would like to be a short fin mako too.
Yeah, and not to forget good genetics, very important.
Good genetics, yeah. I'll have to think about that moving forwards now with my answers. But Mahmood, thank you so much for taking the time to come onto our podcast. We very much appreciate it, and it's been so much fun. I feel like an hour went very fast. That was a whole whirlwind of genetics and interesting and fascinating stuff.
Well, thank you, Isla. It was great chatting with you, and hopefully I was able to make the genetics parts a little more understandable and interesting.
Absolutely, absolutely.
Well,
yeah.
Thank you. Yeah.
Thank you. And just like that, it's the end of the episode, and I think it's definitely fair to say that we learned what we can learn from a shark's DNA. Thank you so much to Professor Shivji for taking time out of his busy schedule and for all of his knowledge and wisdom. We very much appreciate it. And as always, you can find links to everything we discussed in this episode in the show notes on the World of Sharks website. If you like this episode, please be sure to rate, review and subscribe if you haven't done so already. It really means a lot to us, and it also helps more people to find us and find out how amazing sharks are, and who doesn't want that. This was a podcast brought to you by the Save Our Seas Foundation. My name is Isla. I produce and host the podcast. Our amazing visuals are by Jamie Silva. Our lovely logo is by Nicola Poulos, and the wonderful jingle you can hear right now is by David Knight. If you would like to find out more about the Save Our Seas Foundation, you can do so by heading to www.saveourseas.com, or on social media. We are at Save Our Seas on Twitter and at Save Our Seas Foundation on Instagram. And thank you at home for listening. I hope you have a jawsome 2 weeks, and we will see you next time.